session. So next, let me introduce Dr. Rebecca Racz, so our today's keynote speaker. So Rebecca is currently a senior scientific advisor at the U.S. Food and Drug Administration, FDA. She's actually in the specifically FDA's Office of Clinical Pharmacology. So she currently leads multiple projects in the office, including knowledge management and process improvement initiatives. Dr. Racz is a pharmacist by training and received her bachelor degree in Biomedical Science 2013 and PharmD in 2016 from the University of Michigan. She joined the FDA's Division of Applied Regulatory Science in 2016 and was the primary investigator on multiple research projects focusing on secondary and safety pharmacology, natural language processing, and database development. She has published over 30 papers, serves as the lead for numerous international collaborations, and won the 2021 FDA Outstanding Junior Investigator Award. So here I would like to introduce Rebecca. So her talk today is titled Application of Ontology at the U.S. Food and Drug Administration, a use case for secondary pharmacologist. So she will show how ontology and control terminologies can support some uses at the U.S. FDA. So without further ado, Becca, your turn.
Thank you so much, Oliver, for that wonderful introduction. Can you all see my screen?
Yeah.
Perfect. All right. Well, thank you so much for having me here today. As Oliver mentioned, I am Becca Racz. I am in FDA's Office of Clinical Pharmacology, and today I'll be talking to you about the application of ontology at the U.S. Food and Drug Administration, and specifically a little bit about my research in secondary pharmacology. Here is my standard disclaimer that the views presented here are not those of the FDA or HHS. So brief outline. First, I'm going to be going over brief background and introduction to the use of controlled terminologies and ontologies at FDA, and give a couple of examples of the ways that these are used here. And then, as I mentioned, I'll be diving specifically into my research on secondary pharmacology and how ontology and controlled terminology might be useful in that particular field. So first, a brief introduction to FDA data standards. FDA receives hundreds of thousands of submissions every single year in a wide variety of formats. If you can think of some type of file extension, we probably receive it in some way. So in order to simplify the regulatory review process, FDA requires that certain submissions meet data standards, which is a set of rules on how certain types of data are structured, defined, formatted, or exchanged between computer systems. And by meeting these data standards, this really helps reviewers easily find and access the information that they need for review. And it can also support FDA's research efforts. A lot of people think that FDA just does regulatory review, but we do have a lot of research that goes on in the FDA. And we do utilize a lot of the data that we receive in order to try to gain more insights into what is actually being done in drug development and how we can further support that. So at FDA, we do have the FDA Data Standards Advisory Board, which coordinates the evaluation, development, maintenance, and adoption of health and regulatory data standards. So here are a couple of examples of data standards that are at my center, the Center for Drug Evaluation and Research. The first is the ECTD, or Electronic Common Technical Document Standard, which is the standard method for submitting applications, amendments, supplements, and reports. So that's really the big one that industry is using in order to submit their data to us. We also have study data standards. So this is a standard way to exchange study data, non-clinical, clinical information. We do also have standardized product labeling submissions. So those package inserts and drug labels that healthcare providers and sometimes even patients use, that is a standardized format with standardized terminologies. And then additionally, post-marketing safety reporting standards to submit adverse events and periodic reports to the FDA. So we do use controlled terminologies and ontologies at the FDA to receive and analyze this data. And a couple of examples, we do use MEDRA to describe adverse events, particularly those that are received post-marketing. Our database, the FDA Adverse Event Reporting System, receives a lot of information every single day from patients, healthcare providers, lawyers, you name it. We've got it in there. About things that are happening with drugs and other sorts of medical products that are on the market. And so we receive all of that data standardized in MEDRA terminology so that we can easily search and analyze this information to see if there is some sort of labeling change or safety issue that needs to be addressed. We also use SNOMED CT to describe indications for drugs and medical products. We also use structured product labeling, as I mentioned, which is a document markup standard containing 42 sets of control terminologies so that we can issue the product label in a standardized, harmonized way, and it's easily understandable to people. We also have UNIS, which is an FDA-created identifier for substances. This is currently done by the Global Substance Registration System team. The GSRS, Global Substance Registration System, is a huge database with every detail that you can think of for drug products, for applications. A lot of this is actually accessible to the public. There is a public-facing version of GSRS, but we also have one behind the scenes just at FDA that we can access that contains all of the proprietary information. And so in order to maintain this database, GSRS uses a lot of control terminologies, ontologies, standardized identifiers in order to make this information easily accessible. And so they do create and maintain the UNI system for all different types of substances like molecular targets, drugs, you name it, it's probably got a UNI. And then finally, we do use medRT to describe pharmacological class. However, some submission documents still do not come in a standardized format using harmonized language, and this can make regulatory review and research very challenging. And so this is my experience with secondary pharmacology, which does not currently come in a standardized mechanism or with standardized terminologies. So secondary pharmacology or secondary pharmacodynamics are the studies on the mode of action and or effects of a substance not related to its desired therapeutic target. So you can see in the image over on the right that a drug will typically target one, maybe two primary targets, and those are responsible for the efficacy of that particular drug. So when it's known to bind at that particular spot, and when it does, it lowers your blood pressure, helps with your diabetes, whatnot. However, drugs can be pretty promiscuous and can actually go off and bind to a lot of other targets that might be related to the primary target or might be completely unrelated to the primary target. And so these off-target interactions are called secondary pharmacology. And sometimes these could be beneficial. Maybe they help enhance some of the efficacy depending on its relation to the primary target and the downstream effects of the primary target. But a lot of times these effects can be deleterious and cause side effects. So compounds are typically screened against many targets very early in drug development. And this includes receptors, enzymes, ion channels, and transporters that are not the primary mode of action. These screens consist of binding and or functional assays. A pretty typical method is for companies to do the binding assays first. And again, these are done very early in drug development. This is one of the first tests that are actually done on a potential compound. So they'll throw this drug in with around 80 to 100 targets and see what's actually lighting up on this screen. And then whatever comes back as a potentially positive interaction between a drug and a target, they then move on and use functional assays to better characterize this potential interaction. So determine if it's an agonist, antagonist, IC50, things like that. So these screens can help identify potential safety issues very early in drug development by defining the pharmacological profile of a compound. So like I mentioned, a lot of times the primary target is pretty well known, pretty well characterized, but these secondary targets can lead to safety issues. And so by knowing more about where the drug is binding and why, they can better understand any potential safety issues that might come up. And so also they look into the compound's affinity and mode of action at the targets of interest to better understand how this drug, the interactions at this particular drug might lead to some sort of safety issue. So the regulatory assessment of secondary pharmacology is often done by our center's pharmacology and toxicology staff. And this assessment is made in conjunction with data about clinically relevant concentrations and drug exposures to determine the actual safety risk. So for example, if a drug, you go through and do the binding assays, you do the functional assays, and you get an IC50 back, they check that IC50 against the clinically relevant concentration. Because if the clinically relevant concentrations are much lower than that IC50, then you might not have to worry about any potential safety issues because you're never going to reach where that binding is actually a concern within the body. Additionally, the limitations of secondary pharmacology are also taken into account. This includes the selected targets and the extent of testing, as well as the format of the assays. Currently, sponsors are not required to submit any type of justification or reasoning as to why they picked the targets that they picked. A lot of times, sponsors will go through and use one of the kind of traditional safety panels that are available. There's a couple different panels that are well known to address safety issues. So they look at the serotonin receptors, dopamine receptors, opioid receptors, the ones that are commonly responsible for safety issues. But sometimes sponsors might just kind of randomly pick some targets too to see, hey, we think it might bind here, or we're particularly interested in cardiovascular effects, or something like that. But we on our end don't actually know why those targets were chosen. So you kind of have to take it a little bit with a grain of salt. And so based on this assessment and other submitted data, recommendations are made for non-clinical and clinical studies to evaluate and reduce risk. So we were very interested in looking at the data that FDA has on secondary pharmacology and how it could be used. So we evaluated the secondary pharmacology assays that FDA currently has stored in the electronic document room. So we used an R script to identify INDs containing at least one file in the secondary pharmacodynamics folder in FDA's EDR. And this data all currently comes to us in PDF right now. So all of the PDFs were then manually evaluated to identify in vitro data for off-target activity. We had 3 inclusion criteria. The first one was that it needed to be a small molecule compound identifiable in the investigational brochure. And the reason we had this as a criteria is because when these reports come to us, the compound usually has just one type of identifier, just random series of numbers, letters, because again, this is done very early in drug development, so they don't have like a name or necessarily anything like that for the compound at that point. And a lot of times these come in with multiple compounds in a single report. And so these other compounds could be other drugs that they were interested in investigating at that particular point that just never really made it past the initial secondary pharmacology screen or other early drug development tests, or it could be metabolites. And so we need to make sure that we know exactly which compound we're looking at, which results we're looking at, make sure it actually matches the one that's in the investigational brochure, and it wasn't just some random compound that was screened as part of this whole drug development process. We also wanted it to be reported as the % inhibition of control-specific binding and tested at 10 micromolar. Both of these are pretty standard methods of looking at and reporting the data for secondary pharmacology, but there can be some deviations. So in order to kind of look apples to apples at all of these different reports, we included those as criteria. Additional data was harvested from the Global Substance Registration System, or GSRH, which is the system that I talked about a little bit earlier, where they actually make the Unicodes. So from there, we looked at the compound name, sponsor status, submission year, and indication. And we also gathered some safety information from the daily med drug labels for the ones that were approved. So in total, we extracted about 2,700 INDs that had at least one file in the secondary pharmacodynamics folder. And of those INDs, about 1,100 contained at least one file of binding assay data that met the inclusion criteria. And omissions primarily occurred because there was either no binding information, like the raw data, maybe it's just a summary of the study, but it didn't actually have the numerical values in there, or the data was reported in a format other than % inhibition. So the submissions were from 480 sponsors from 1989 to 2020. And the top 3 most common therapeutic areas were oncology, neurology, and psychiatry. 60.5% of the IND applications were active, and 52 submissions were associated with an approved new drug application. And this is as of 2020. So I'm sure several more have been approved since then. But we initially grabbed this data in 2020. So we did notice that the percentage of INDs containing secondary pharmacology has actually increased in recent years, which is great. Right now we're hovering around 18-ish% of INDs have secondary pharmacology data that's actually submitted. Typically, it's pretty standard for companies to actually conduct these secondary pharmacology studies, but it doesn't always get submitted to us for evaluation. So it's very exciting to see that companies are submitting more and more of this data so that we can try to make early decisions on safety concerns very early on in the regulatory review process. In total, we had 747 distinct targets. There were anywhere between 1 to 322 targets per report. The mean was 74 and the median was 69. We also had 1 to 938 reports per target. The mean was 110 and the median was 23. So that's what you can see over here in this graph on the left, is that we had a couple of targets on the bottom that were tested a whole bunch of times. And again, those are going to be kind of your standard safety concerning targets like dopamine, serotonin, and opioid that are very commonly associated with adverse events and are part of those more standardized panels that I mentioned earlier in the talk for safety events. But we did have a lot of targets that were tested just a couple of times. And so that's going to be on the top of the graph here, the little peak to the funnel. And so again, those are targets that were probably specifically chosen by the sponsor for a particular reason. But since they are not required to submit the reason, we are not sure why they specifically chose those targets. However, you can see on this graph that the targets that were specifically chosen so that they weren't tested that many times, you can see over on the left-hand side of this graph, those often had a pretty high hit rate. So they were obviously chosen because the sponsor had some sort of feeling that their compound was going to interact with that particular target. We don't know why they thought that, but usually they were right because the low testing rate often had a high hit rate. There were a few notable examples, exceptions, including BMAT2, melatonin receptors, sodium and chloride channels, and adenosine targets. And then enzymes were actually more associated with a low testing rate and a high hit rate. We did look at the correlation between the number of labeled safety events for the ones that were actually approved, the approval of an associated NDA, and the status active or inactive. We did not see any correlation between the testing and hit rates and those results. So this study is limited because we did only include applications submitted to the FDA. This is typically going to be cleaner data and cleaner compounds, because anything that really lights up a secondary pharmacology panel is probably not going to be pursued much further, because that indicates there might be some potential safety concerns. And there may be some data outside of the secondary pharmacodynamics folder. So the study we started finding data from 1989, and this is very early on in the electronic document room. Things were, kind of messy, weren't necessarily always stored in the same way as it is right now. Now it's very regulated and very tight how the data actually comes in and is stored. So it's very possible that we did miss some early secondary pharmacodynamic studies because they could have been placed in other places in the electronic document room. But our main challenge was really the lack of harmonization in terminology and file structure. So first of all, targets were listed with a whole bunch of different names. There's a lot of target synonyms out there that are known, but there were also targets that were listed as 5-HT2B- company name. And we don't know if that's actually the same thing as 5-HT2B, if that's some sort of, did they change the structure of the particular target so it can't be looped or lumped together with 5-HT2B. So that was actually very challenging and very manual work to try to actually condense all of the results for the different targets, because we didn't quite know if those were the same targets, if they were different targets. So that was really one of our big issues. Additionally, as I mentioned earlier, the code name and the assay results needed to be confirmed in the other submission documents, because they just kind of assigned some random numbers, letters, very early on in the drug development space, but it doesn't necessarily match up with the name that we now know it as, the generic name or whatever identifier that the company gave it as it started to go through the drug development process. So we had to go back and manually confirm that information in the investigational brochure, which was definitely time consuming. Additionally, the table structure could differ significantly between different CROs, different pharmaceutical companies, which made automated extraction very difficult. So like I said, this data all came in PDFs, and so we are trying to use some different PDF reader tools in order to actually extract this data and put it into an Excel file. But even like the column names were different, the columns were in different orders from each other. And so it made it very difficult to actually line this all up. And so that's why this turned into a very much manual process. So in order to try to address some of these challenges, we did enter a public-private partnership with the Bestoya Alliance in June of 2022. And the goal of this partnership is to harmonize in vitro pharmacology submissions, including secondary pharmacology. There is a website that you can look at and see a little bit more about what we're doing in the IVP, in vitro pharmacology working group. But right now we're working on trying to put together an ontology and a harmonized way to actually look at targets, look at the assays and the assays format, how to actually submit this data and get it ingested into FDA's GSRS system, and also, make sure that we're providing enough information for the regulatory review process as well. So it's been very exciting to actually participate in this project and make sure that we can get kind of a harmonized data standard and way of submission moving forward, so that this information can be used a little bit easier in regulatory review and research. So in conclusion, FDA extensively uses controlled vocabularies and ontologies in the regulatory review and research processes, but there are still areas where implementation of these tools could be beneficial, like secondary pharmacology. So harmonization of secondary pharmacology can enable the generation of mechanistic hypotheses and inform regulatory decision-making. And further research is needed to determine the value of secondary pharmacology in predicting non-clinical and clinical outcomes. As you may have heard, there's a lot of interest right now in novel approach methodologies or NAMs. And so we're definitely interested to see how some of these these in vitro type of assays can actually help support our mission to reduce, replace, and refine animal testing. And so we're hopeful that with more standardized submission of things like secondary pharmacology, we might be able to draw more conclusions between some of these very early screenings and the actual non-clinical and clinical outcomes. And that might help us address the 3 Rs. I'd like to acknowledge Andrew, Christina, and Marianne, who are they were my ORISE fellows who did most of the work here. It was a big lift trying to automate this, and then finally kind of turning to the manual extraction of most of this data. And so all 3 of them did a great job. And then I'd also like to acknowledge Kevin Snyder, who was in the FDA PharmTalk staff. He's now at Certara. But he was really kind of the point of contact to make sure that all of the information we were gathering was still relevant to regulatory review processes as well. So thank you very much for having me here today. Thank you very much for listening, and I am happy to take any questions.
Great, Becca. Yeah, I learned a lot actually from you. So thank you for your wonderful talk.
Absolutely.
Let's see. Seems like we are having many questions. I have my own question, but before I can ask, let's see. Let's Asia, you ask a question, right? In the chat. Do you want to speak out?
Yeah, can you hear me?
Yeah.
Oh, okay. So my question is more, actually I have a lot of questions, but also, yeah, thank you, Rebecca. I learned a lot from you also. So one is that you mentioned in the SPL, there are 42 controlled terminologies. So where can we find those names of those controlled terminologies? And then
oh, sorry, go ahead.
Yeah, the second one is, so there are a lot of FDA data standards. How does it compare to the EMA standards and international? Are they aligned, or they all use the same standards? What's the dynamics there? Can you introduce? Yeah, thank you.
Yeah, so hi. It's nice to see you, Asia. As far as your question on the 42 controlled terminologies in the SPL, I have a website linked at the bottom of my slides. I can also pop it in the chat. And that lists a lot of different information on the FDA data standards and the different types of controlled terminologies that are used. So I am sure it's on that website somewhere. I'll see if I can find it real quick too and pop that in the chat. As far as the alignment with EMA, I am not a data standards expert at FDA. We do have a data standards team. And so I'm happy to reach out to them and see if they have any more information that they can provide about that and follow up with you.
Thank you.
Absolutely.
Great. So maybe Becca, I can ask one question. So I see you have talked a lot about the secondary pharmacology, right? So are they all about chemical drugs, or you are also thinking about vaccines, for example, or immunotherapy methods as well?
Yeah, great question. So since I work in CEDAR, our primary concern is with with drugs. A lot of them are small molecules, especially. Small molecules are very frequently tested in the secondary pharmacology space. The larger molecules, the biologics, and some of these other immunotherapeutics, and the vaccines, some of those are regulated by CBER, and I know that they have their own program currently that's looking into secondary pharmacology, but I think that the methods that are used over there are very different than what's typically used in small molecules. So I personally am not quite sure what's going on as far as like vaccines and biologics, things like that. But I am happy to look into that a little bit further. I do think that there was a presentation last year on what CBER is doing in secondary pharmacology. So I'll see if I can find that.
Great, yeah, thank you. So let's see, for the small molecules, my other question is like, on average, how many secondary targets you can find for any single small molecule?
So the small molecules were typically tested around like I think it was about 70. Let me look back at my slides real quick. They were tested at a mean of 74 targets. So each individual small molecule was tested at about 74 targets.
I see. So the 74 is more like the standard list, right? But it may or may not target on any of the 74 targets.
Exactly. Yep. So there is a standard list out there. It's called the VDOS2025, and this was a paper that was published in 2012. And so those 44 targets are very commonly chosen for screening in secondary pharmacology. So like I was mentioning, those are kind of like the traditional safety targets, like the opioids, the serotonin, things like that, the dopamine. So a lot of times people will pick the 44 and then they'll pick some other targets. We as FDA don't get any information about why they chose those, other 30-ish targets or so, but there's definitely some sort of reasoning behind the sponsor. It's not just kind of like throwing a dart at a dartboard and choosing targets. But yeah, they could, the compounds could bind to all of them, or they could bind to none of them. And we actually have a lot of screens that have just completely negative data, which in the end is good, right? Because hopefully they're not associated with too many safety issues.
Great, thank you. So let's see, are there any other questions from anyone?
There's one question from Yuki in the chat.
Oh yeah, Yuki?
Yes. Thank you for your great talk. And I have one question. In my previous work, I worked on developing toxic process ontology for liver hepatic toxicity. So I'm curious how the FDA try to integrate ontology work for mechanisms for toxicity.
So to my knowledge, we don't actually have any connection between the secondary pharmacology targets and then the adverse outcomes, the adverse clinical outcomes, and the like the actual mechanisms. I don't think we have any direct connection, but I think that's something that we're hoping to establish at some point, because as I mentioned, we're very interested in the novel approach methodologies, the NAMs. We'd really like to see if we could use secondary pharmacology for something like this to actually say, okay, your day one of drug development, we see that you're hitting these 5 targets. What do these 5 targets eventually mean in your clinical studies that you're going to do in, a decade or whatnot? So that is definitely something that we're very interested in connecting together. Unfortunately, we just don't have enough data right now, but we're hoping that with a more standardized submission process, we might be able to actually like make that connection between target and adverse events, and then also make a connection between, potential mechanisms. Like we see, okay, you hit this target and we know here's, the downstream effects of this target. So maybe there's not a direct connection, but, it might eventually lead to these types of outcomes. Or additionally, we see that you're hitting this whole cluster of targets, and this whole cluster of targets might be more responsible for this particular outcome. So definitely something that is of interest.
Yes, thank you.
Absolutely.
Yeah.
Great. Okay, so any other questions from anyone? Yeah, so we have 2 more minutes. So maybe I can ask you another question. So
Sure.
Okay, so just trying to see how we can link the 2 together, right? How can we link ontology or control terminologies to the mechanistic study of the secondary pharmacology phenomena? So one thing is you can do the standardization, right? I'm kind of thinking, and any other things can we use the ontology to also support some data analysis as well?
Yeah, so yeah, that's kind of the plan at this point, is that once we get everything, like the actual submitted data, harmonized, standardized, and so we're receiving all of the targets and all of the data in a very standardized format. That does, that lends itself to kind of some of these more standardized formal analyses, because then we could connect, say we have like this particular target, we know it links to all of these other linkers in all of these different databases. Like we have like the Hugo nomenclature for targets and NCBI, like we have all of these different databases, and all of these databases have different types of information, right? And so we might hopefully be able to connect these targets and these results with, okay, here's, maybe a KEGG pathway or, some other type of mechanistic pathway. And then we can just kind of like start spreading out the data. And then, we eventually get the clinical data in too, and the non-clinical data that we can map to other types of ontologies, control terminologies, like MedDRA, for example. Hopefully by having all of this data standardized, I think it will make it a lot easier to try to manipulate the data and actually match it to all of these different mechanistic pathways, these adverse events, these clinical outcomes, non-clinical outcomes. And so I think we'll be able to get a lot more use out of this data once it's all harmonized.
Great, wonderful.